Hello, everybody. I'm Paolo de los Rios. I'm the head of the Laboratory of Statistical Biophysics here at EPFL. And today I would like to just give you a brief introduction to the research that is taking place in my laboratory. Of course, it's called laboratory, but it is actually a theoretical laboratory. So I'm doing theoretical physics. Let me give you a glance of my scientific path. First of all, by master, I'm an engineer, as most of you are. Took a master in electrical engineering in Torino, at the Technical School of Torino. Then I moved for a PhD in theoretical statistical physics in Trieste, which is in the eastern part of Italy, of northern Italy. And from there, then I moved for a Max Planck, for a postdoc at the Max Planck Institute for the Physics of Complex Systems in Dresden, in Germany. And then that's where I came after that in Switzerland for a postdoc at the University of Fribourg. And after that, I finally arrived in Lausanne, taking sort of almost a full circle. And I've been first at the university, then at EPFL, and I'm an associate professor since 2010. My activity, my research activity has mostly two axes. First of all, it's in biophysics. It's strongly biased toward biological problems. And one of them is how to analyze biological non-equilibrium processes in the cell. I will come to this a little bit more, and in particular chaperone proteins. And the other one is how to use machine learning, because of course some new techniques, including neural networks that have become pretty popular in recent years, to explore the relation between the structure, the sequence, and even the function of proteins. So let me start from non-equilibrium processes in the cell. Of course, we know cells need to consume energy to work. Without food, and thus energy, every cell dies. Essentially, the idea is that this is needed to work against entropy, to work against the natural tendency of systems to reach their minimum of the free energy, which is the equilibrium in thermodynamics, which is, of course, death. And just to give you two examples, DNA duplication. We know that cells, when they divide, they need to replicate their DNA. And if we just would use a simple basic thermodynamics, essentially energies, forces, and whatnot, every about 100 bases of DNA that are duplicated, there would be a mistake. So think about the fact that we have of the order of magnitude of 1 billion bases in our DNA. This would mean that we would have about 10 million errors every time a cell duplicates. After 100 duplications, the DNA wouldn't be the same. Instead, the error rate is of the order of 1 every 10 to the 9, even less bases. So clearly, thermodynamic considerations tell us that this is impossible unless thermodynamics is defined, equilibrium thermodynamics is defined by consuming energy. Meaning there are proofreading processes that allow this duplication process to be much more reliable than pure thermodynamics, equilibrium thermodynamics would predict. This is not surprising. It's like when you try to correct errors, when you write, you have to spend energy to cross-check, go back, reread your text, and so on. So it needs energy, it needs to perform some work on information, but it's still work. But another case is transport across membranes. We know that, for example, for many substances, there is a clear gradient across the cellular membrane with concentrations, for example, of drugs, of poisons that are typically much lower inside the cell because there are pumps that that throw away these noxious molecules, and nutrients that concentration inside the cell is typically much larger than the concentration outside. So this, of course, defies equilibrium that would require the same concentration inside and outside. There are machines, the proteins that consume energy to actively transport molecules, nutrient or drugs and others across the membranes. So what we try to do in the lab is to tie in a very precise way the function of proteins of these machines, of these molecular machines, to their energy consumption. And one prime example that we look at are chaperone proteins. Just as a reminder, chaperone proteins are proteins that help other proteins fold correctly, because the folding problem, that is, once a protein is produced by the ribosome, it will try to fold into its correct, three-dimensional correct and functional form. Oftentimes, this process can go wrong, and if there is no intervention, then diseases can emerge, like Alzheimer, Parkinson, amyotrophic lateral sclerosis, and so on. So there are many diseases that are related to the problem what is called the misfolding, that is wrong folding of proteins. This is a very much physical problem that is intrinsic in the nature of proteins. Nature has evolved several machines, collectively called chaperones. You can think that the word already tell you what these chaperones are about. They supervise the behavior of other proteins. These chaperones use ATP and they steer the folding of other proteins in the correct direction so that the folding will be easier and more reliable. And they are very fascinating machines that are getting more and more interest in the community. And we work together with experimentalists also to try and understand how these chaperones optimize energy consumption to turn the energy that typically comes from ATP into profitable folding proficient work. So this is the part of non-equilibrium processes in the cell. Then we have also another part which is unrelated, although maybe to some extent related to the folding problem. As I say, the folding problem can be complicated, but what is interesting is that when a protein folds and can do so autonomously in solution, will acquire a certain three-dimensional shape when everything goes right, otherwise chaperones intervene, and with a precise function. And the fact is that, as you know, evolution implies that there are mutations from one cell to the other, from one organism to the other. And yet, oftentimes these organisms contain the same, in quotes, protein. Same in the sense that it's the same, recognizably the same protein that falls into the same shape and performs the same function. So the question is then, if they can do so autonomously without the help of any other proteins, apart possibly from chaperones. This means that the rules to fold and to function are written in the sequence of amino acids. And the fact that you can write, nature writes these rules in so many different ways, tells that there is like looking at a language, at the same sentence written in different languages. If you have a sort of a Rosetta Stone, you can try to use all these sequences as a sort of a Rosetta Stone to crack the code of folding. This is precisely what maybe you've heard about AlphaFold, the program that is able to fold proteins does. And so what we are trying to do is to use machine learning algorithms that are very powerful, among them neural networks, but not only, to unveil the relation between the sequence, the structure, and the function of proteins. Part of it is just pure fundamental science, but one might think that at some point this might help also, for example, to design new proteins. There are other groups at EPFL that move along this direction. And one can try to understand what evolution has learned about physics and how evolution has organized all these protein families. So these are, just to summarize, the two lines of research that we have in the lab. So if you're interested, of course, most welcome to contact me by email, and we can organize some meeting. Thank you, and goodbye.